So whenever you're ready, you're welcome to begin.
Hello, I'm Michael Margolis. This is Anissa, that's Gaia, and that's Victoria. And I'd like to present to you our D-microvirus project with the Torrey Pines CCA San Diego iGEM team. And we engineered E. coli capable of extracellular secretion of mycotoxin detoxifying enzymes. Our project mainly focused on mycotoxins. Mycotoxins are fungal secondary metabolites that are present in 20% of all crops, like maize, cereals, coffee, and nuts. Mycotoxins are, of course, extremely toxic as well. They have 68 times the toxicity of arsenic, and they're widespread. According to the World Health Organization, they're the third leading cause of cancer, and there's over 600,000 new cancer cases per year related to mycotoxins alone. And a large number of these cases occur in developing nations, where they can't properly store the crops listed above. Because of improper storage, the fungi that produce the mycotoxins are more likely to grow, and therefore more mycotoxins are present on the foods. Our project focused on two mycotoxins, aflatoxin B1, produced by Aspergillus flavus, and zevalonone, produced by Fusarium graminearum. Aflatoxin is a class 1 carcinogen, which means that it's been studied in humans and is known to be carcinogenic. It's also deadly if inhaled, ingested, or if it comes in contact with skin in even the smallest doses, making it the deadliest of all the mycotoxins. We also studied zevalonone, which is an estrogen mimic that can cause growth defects. We found that certain fungi, like Amarelliella tabascens, actually produce enzymes that can take apart these toxins. So aflatoxin B-detoxifizyme, or ADTZ, is produced by Amarelliella, and it's 77 kilodaltons in size and is difficult to extract from the original fungus, which is why the bacterial system we used is especially useful. Here we see the ADTZ detoxification pathway. This highlighted part of the aflatoxin, the epoxide, binds to a guanine in the P53 gene, which is well known to be a cancer suppressor gene. Because of this binding, it prevents P53 from properly working, which makes it extremely carcinogenic. However, ADTZ, through a simple hydrolysis of the epoxide, changes the aflatoxin into a much less deadly form. We also looked at Clonostatus rosea, which produces ZHD101, or zevalonone hydrolase. This is only 29.2 kilodaltons in size, and it's also difficult to extract from the original fungus, which is why we used our particular bacterial system. And here we see the ZHD101 detoxification pathway. Zevalonone, which is an estrogen mimic, can be just cleaved into a non-harmful form.
So the main idea for our project is to utilize enzymes that are naturally found and produced in fungus, and then to take them and design recombinant DNA for E. coli. So we chose to go with unoptimized cDNA for ADTZ, and this was because in a previous study they used unoptimized cDNA and expression was successful. For ZHD101, we chose to go with optimized cDNA, so we have the best chance of getting the single peptides that we want and for getting the correct amino acids. We can also do a comparison between the optimized cDNA and the unoptimized cDNA. So the unique part of our project was attaching a secretion signal peptide at the N-terminus of the protein. And this was important because in fungus, they're produced intracellularly, and they're also stored there. So to lyse the fungus would be extremely not good for cost. And also, if you were to design a system in which you had a bacteria that produced these enzymes intracellularly and didn't secrete them, it'd also be bad for cost. Also, by having the secretion signal peptide, we can have mass commercialization, mass production, cheaper production, and you can spray them directly onto crops you think might be infected or crops you know for sure are infected. And there's no cure, so prevention is the best cure. So the next thing we went on to do was look at secretion pathways, and we focused on two, SEC and TAP. The first one we looked at was TAP. This is the twin arginine translocation pathway, and it translocates folded proteins into the cytoplasm. There were a couple of reasons we chose not to go with this one. It's mostly for proteins that have a tightly bound cofactor, which does not apply to us. It also has poor export efficiency, and there tends to be accumulation in the translocons. So the next one we looked at was SEC, and then this one is the pathway that secretes unfolded protein into the periplasm. So for most secretions, it does create functional proteins. It's also likely to have correct folding because it has chaperones. This minimizes degradation, and it forms correct disulfide bridges and slows down translation, which can help with the problem of accumulation. The next thing we went on to do was look at signal peptides. So we looked at ones that were SEC dependent. So we chose two, alpha amylase and beta lactamase. And for alpha amylase, there is natural secretion of alpha amylase in E. coli. And this is good because we have a higher chance of our protein actually secreting. It's also suitable for E. coli strain K, which is the strain we chose. And like I said, it's SEC dependent, which is the pathway we chose. So the second one we also went with was beta lactamase. This one is also secreted naturally by E. coli, which is good because we'll have a good chance of our protein being secreted, and it's especially prevalent in an environment with antibiotics, and our vector has AMP resistance in it. Also, the signal peptide had succeeded in exporting fusion proteins, and it's good for strain K, which is the strain we chose, and again, it's SEC dependent, which is the pathway we chose.
So now we move on to the schematics of our project. So first we chose the vector to use. We chose the PTRC-PIS-A vector because it has the TRC promoter. TRC promoter is a synthetic promoter of TRP and LAC. And it is decently strong, and it also is known to be pretty good for secretion as well. It also has enhanced translation with the mesostrons, and it has the PIS tag, which we can use later on for further verification or detection, for example, by Western blots. It also has the AMP resistance, which, as Anissa mentioned, may help us with the beta lactamase secretion. And even though this copy is actually a mediated copy number plasmid, we decided this on purpose because if we chose a high copy and we don't know the level of secretion of the proteins that we're expressing yet, it might lead up to accumulation problems. So we moved on to synthesize the fragments that we want. We divided ADPZ into 3 fragments because it was pretty long, but we just chose one fragment as, we synthesized one fragment for GHD101. And we PCR amplify them to have the tails they want because we use seamless cloning. Seamless cloning is a method of assembling different DNAs together, especially when it has around 15 pairs of overlaps. What it does is several exonucleases kind of chew back on the 5' ends of the DNA. And for example, if you zoom in to this junction right here, it looks something like this, where the 5' ends are cleaved away, not cleaved away, chewed back. So it actually forms something like a sticky end which will attract. And if you just transform this right away to E. coli, the ligases in E. coli will actually just fuse the backbones together for you. So it just becomes a full plasmid by itself. So these are the 4 final constructs that we designed. The first one, as you can see from here to here, is just the vector. And right after the start codon, we have the signal peptide, and after that we have the gene of interest. And we have this PIS tag after the gene because we want to put it in the C terminus instead of the N terminus because there are a class of proteins called signal peptidases that actually cleave away the N terminus and cleaves away the signal peptide. So these 4 are different combinations of the 2 signal peptides that we chose and the 2 genes that we chose. And just AZ refers to, AA refers to alpha amylase with ADTZ, AZ refers to alpha amylase with GHD101, and for BA and BZ, similarly for beta lactamase. So from here we go into the results. This was just a quick verification step to kind of see that we made the right fragments, amplified them right. And we did indeed, so we column purified them and used them for seamless cloning. And right after seamless cloning, we transformed them and we had over 10 colonies for each type, which was good. But we also wanted to make sure that we had the right colonies. So we went on to colony PCR, and we were able to narrow down to the colonies that seemed to have the right size of the inserts. But we also wanted to be extra sure, so we sent the plasmids to sequencing, and compared the published cDNA of the proteins to the sequencing results that we got. And it turned, unfortunately, BAs and BZs, we weren't able to proceed with those because, as you can see here, they usually had point mutations, either addition or deletion that completely messed up the frame. But also, this was not surprising because it is a synthetic genetic gene, and it is rather common to have mutations. So we moved on to the AAs and AZs, which had a 100% match. So what we did was we cultured the AA and AZ colonies, split them into half, and induced half of them with IPTG and the other half with nothing, because they do have a lag-inducible operon, and we could just compare those two to kind of verify expression. And after that, we spun them down, separated the pellets and the supernatants, because if they are excreted extracellularly, the protein should be in the supernatant, and compared the pellets and the supernatant to kind of verify secretion. So this is a quick prediction table that we made. As you can see, the green part is the one that we want. The first column is where the IPTG-induced cultures will actually have more of the proteins that we're looking for. And the first two rows refer to the supernatants having the proteins that we want. But ideally, we would want the middle row because it has the few protein in the pellet and a lot in the supernatant, which signifies very efficient secretion.
So before we started with the page gel, we had to do some quantitative matching so that when we ran the samples, we'd be able to compare the relative band sizes or the relative band thicknesses to see the amount of protein in the pellet and the supernatant. So in the pellet, if we started with X unit of proteins, after spinning it down and resuspending in SCS loading buffer and adding 2 microliters for our sample, we ended up with X over 100 unit of proteins. And similarly in the supernatant, after reconcentrating the supernatant and using 5 microliters of the SCS loading buffer plus the reconcentrated, we also ended up with Y over 100 unit of proteins. So here's the SCS page gel number 1 that we ran. As you can see, the supernatant bands were not dark enough to show up. That's because our concentration was not high enough for the SCS page gel to show it. And also there wasn't much noticeable difference between the induced and uninduced pellets. So this page gel, we could not confirm expression or secretion from this page gel, so we proceeded with Western blotting. And in Western blotting, we used the chromogenic method, which is a bit different than the chemiluminescent method in the way that at the end we add a BCIP pellet, and this BCIP is hydrolyzed by alkaline phosphatase, and it forms an intermediate, which then goes through dimerization and leaves an indigo dye that shows up on the membrane. And so here's our membrane. As you can see in the boxed bands, there is significant darker bands in the induced AZ pellets, and there's also double bands. And the top band is the uncleaved product that's still in the cytoplasm, and the lower band is the cleaved product without the signal peptide in the periplasm. And this Western blot was able to show us successful expression, but we could not confirm excretion because the supernatant still did not show up. And the reason for this was because the chromogenic method is still not sensitive enough compared to the chemiluminescent method. So, and also for alpha A, because of the large size, there's perhaps a shorter half-life, so it could have been degraded. And there could have been a masking effect in which during protein folding, the His tag wasn't exposed enough. So we had to do some troubleshooting after this because we wanted secretion. And this time we decided to take all the supernatants for the sequence verified colonies of alpha A and alpha Z, and we reconcentrated them to even more, so it's times 50 this time, which is much more than we did previously. And then we ran an SCS page gel number 2. And the SCS page gel number 2, as you can see from the far left, is a standard molecular weight ladder. And in the control, we ran just the vector with nothing else added. And then in the box areas, there is the bands from the supernatant that we ran. And alpha A shows darker bands at around the 70 kilodalton area, which is the size that we want for alpha A. And similarly for alpha Z, it's around 30 kilodaltons, and it is slightly darker than the control. So through this page gel number 2, we can confirm potential secretion from the in the supernatant, and because it was secreted, we can confirm expression.
So in conclusion, we were able to sequence verify AA and AZ, and we also verified expression for AA and AZ, as you remember from the Western blot and the SCS page gel number 1. And we also saw potential secretion for the SCS page gel number 2. Now, we also found out that the problem was the concentration of the supernatant, which could mean several things. It could either mean there was secretion, but so little that we couldn't detect it. Either because it's just all stored in the paraplasm, not actually actively secreted out to the extracellular space, to the supernatant. Or it could have been aggregated within the cell or outside the cell, and if you split it down, it would actually be in the pellet area, not in the supernatant. Or there could have been no secretion because of some of the incompatibilities with the secondary structures of our protein and the sickle peptides. But in order to verify those, we also need to have some future corrections. So we should confirm the second page gel with Western blotting, just kind of confirm secretion. And we should also redo the beta lactamase sickle peptides, EABCs, because we never actually got to compare them. We should also try the same expression in other organisms such as yeast or fungi, because they actually do have a more pronounced secretion system, but they are harder to work with. Or we could try other strains of E. coli. We chose strain K because it was published that they were compatible with the signal peptides that we chose. But in some unmodified strains, there are actually inclusion bodies of unfolded proteins that might have interfered with the secretion of our proteins. And we can also do time control experiments because E. coli may actually secrete the proteins in different points of their lifetime. Or we could check periplasmic proteins, because as we said, it might have been all stocked in the periplasm, so we could just use detergents to kind of strip away the very outer layer of E. coli because they are gram-negative. Or we could attach GFP to the proteins to kind of have a solid numeric value of how much protein we actually had. And this is more of a long-term goal, but we could design a functionality assay in which we test how much of the mycotoxins they actually degrade. So just quickly, during our experiments, we only used BSL number 1 organisms. We were also under constant supervision of experienced researchers, and we always wore proper attire, such as goggles, gloves, lab coats, etc. And we are certified by UCSC Environmental Health and Safety Division to be able to work in the lab conditions and have the proper knowledge of safety. So we also had a various human practice project that we want to tell you guys about. First was the collaboration. We were a single high school team last year, so we had some of the experiences, some of the resources, people who are willing to help us. So we actually invited our neighboring high school to kind of join our team so we could brainstorm ideas together, do the lab work together, and have lectures together basically, listen to lectures together. And the lectures were also open to people just not limited to people in iGEM. It was just anyone in the community who actually heard about the lectures or were interested in synthetic biology were able to come and attend the lectures. So it was a community project as well. The second was a video about how to start an iGEM team. You can actually find this video in our wiki page, the outreach section. Since we also had trouble last year just kind of getting things rolling along, so we just listed some tips and guidances about how to form an iGEM team that would help out people interested in making a new team or just new teams in general. For example, how to get funding, how to get people who are willing to help you, how to get donations, with lab space, etc. So we actually emailed this video out to around 300 principals in California and offered them guidance in forming an iGEM team in their school as well. And if distance will allow from our area, we'll actually visit them in person, talk to them, and guide them through the process personally about how to form an iGEM team. This was a major one. So we're actually really affiliated with the Journal of Youth and Science. It's an international science journal for high school students. It's also student run, but it's actually spread to all over the world, like all over the city, all over the United States, Korea, India, Mexico, Turkey, Canada, etc. So what we do is the Journal of Youth and Science is focused on original research. We also, our goal with this is to promote this to other iGEM teams as well, just kind of have a community of young scientists who inspire each other, and just kind of get ideas, brainstorm ideas together, and just tell them what we do as well. So actually, the Mexican team from last year agreed to write an article about their project last year, basically, and it'll be published in our next issue. And all various iGEM teams also have agreed to write up an article about their projects, which will definitely help out the cause. And The Journal of Youth and Science is not just focused on synthetic biology. It also focuses on all the various aspects such as chemistry, physics, nanotechnology, etc. So the idea about this is to kind of get people to have an idea about interdisciplinary thinking. For example, see how synthetic biology can apply to other fields of science, and see how other fields of science can kind of have an application in synthetic biology. Also, the Journal of Youth and Science is not just limited to original research publication. It also has people can also submit articles about just discussion about past works, past research projects. So we actually had many articles that relate to ethics questions, just discussions in general. So we hope to propagate this to other Argentines as well and have it spread the good cause. So thank you, that was our project. We'd like to acknowledge and thank people for being very instrumental in our team success. Yay, synthetic biology!